Welcome to this explainer. We're going to take a pretty wild journey today, zooming all the way from the everyday act of eating food right down to the incredible microscopic molecular machinery that actually makes your survival possible. I mean, we all eat every single day, right? But we very rarely stop to think about the spectacular, ultra-complex biological factory operating completely silently just beneath our skin. Here is our roadmap for today. We're going to start with the digestion problem, look at how we test for nutrients, actually meet the enzymes, see how they work, and then wrap up with the factors affecting their rate. Let's dive in.
All right, part one, the digestion problem.
So I want you to really think about this for a second. How on earth does a massive, juicy burger actually get inside your microscopic cells? Your cells are absolutely desperate for energy to keep you alive, but that energy arrives as these large, insoluble lumps of food. A burger, as amazing as it is, can't just magically teleport into your bloodstream. We're dealing with a massive size and chemical mismatch here. Well, the solution, of course, is digestion. It's the process that safely dismantles these giant molecules into the tiny, soluble products you see right here. It is literally like taking a massive Lego castle and smashing it down into individual bricks just so they can fit through a tiny doorway. Complex starch gets broken down into simple glucose. Massive proteins are unlinked into individual amino acids. And bulky fats, they get snapped into glycerol and fatty acids. It's only when they're in these tiny forms that they can slip into your blood and actually power your body. Moving right along to section 2, testing for nutrients. Before we meet the biological workers doing all this heavy lifting, let's look at how biologists actually reveal these invisible nutrients in a lab. It takes some highly specific chemical recipes. Take glucose, for example. You can't just mix things together randomly. You have to add this special blue liquid called Benedict's reagent, and then, this is absolutely crucial, you have to heat it in a boiling water bath for a solid 5 minutes before you get your result. And here is the really cool part. If simple sugars are in there, you don't just get some random shade of red. In biology, the positive result is strictly defined as this vibrant brick red color. It's a highly specific reaction, confirming you've got the exact fuel your cells use. Now, what if we're hunting for complex carbohydrates, like the starch in a potato? For that, we don't even need heat. We literally just add some iodine. If starch is present, bam! We get this highly specific, dramatic shift from orange-brown to a really deep blue-black color. And for proteins, the stuff building and repairing your muscles, we use the Burette test. You drop that solution in, and you're looking for this beautifully distinct color shift, going from a pale blue straight to a rich violet. So, bringing all of that together, you can see how we use these precise chemical color changes to perfectly identify the raw materials in our food. Blue-black means starch, brick red is glucose, and violet is protein. It's basically a genius chemical fingerprinting system for your lunch. Okay, part 3, meet the enzymes. So who are the microscopic heroes actually breaking your food down? The enzymes. These guys are your body's highly specialized factory workers. By definition, they are biological catalysts. That means they wildly speed up chemical reactions, specifically breaking those chemical bonds in your food without ever being consumed or permanently altered themselves. They do the exact same job millions of times a second. It's wild. And you've got 3 main crews working in this factory. Carbohydrases, like the amylase, right there in your saliva, start breaking down sugars the second you take a bite. Then you've got proteases, like pepsin, tackling the really tough proteins down in the acidic environment of your stomach. And finally, lipases, pumped out by the pancreas, handling all the stubborn fats and oils. Which brings us to section 4, how enzymes actually work. Now, our understanding of this has evolved so beautifully. For the longest time, starting way back with Emil Fischer in 1890, we thought enzymes were completely rigid. We called it the lock and key model, assuming the food molecule fit perfectly into the enzyme like a stiff metal key into a lock. But science didn't stop there. By 1959, Koshland proposed the induced fit model, realizing that enzymes are actually wonderfully flexible. Check out that modern induced fit model on the right. See how the enzyme isn't just a rigid block. As the substrate, the food, enters the active site, the enzyme actually slightly changes its shape to mold perfectly around it. Think of it less like a metal lock and way more like a flexible baseball glove snapping around the ball to get that perfect grip. And by grabbing it in this incredibly precise, snug way, Enzymes do something absolutely miraculous to the physics of the reaction. They lower the activation energy. Look at that massive red curve. That's the energy hill the reaction normally has to climb. But with an enzyme, it drops right down to that blue curve. The enzyme physically strains the bonds in the food molecule, making the whole biochemical process infinitely faster and easier.
Finally, Section 5, Factors Affecting Enzyme Rate.
Here's the catch, though. Our microscopic factory workers are incredibly sensitive. Look really closely at these graphs. Both temperature and pH have a perfect optimum peak where the enzymes work at absolute top speed. But if you push the heat or the acidity just a tiny bit past that peak, the rate doesn't just slow down, it craters. The enzymes literally lose their 3D shape, a process called denaturation. The glove is destroyed and the workers stop entirely. This is exactly why a severe fever or a sudden pH change is biologically so dangerous for us.
It's a similar story with concentration. Add more enzymes to a system, and you get a fantastic linear speed boost. More workers equals faster processing. But notice what happens if you just keep adding more food, more substrate. It only speeds up until it hits a saturation point. At that plateau, every single enzyme worker is 100% busy. You can add all the food you want, but the microscopic factory is totally maxed out, and the reaction rate just flatlines. So as we wrap up this explainer, I want to leave you with this to ponder. We've seen how insanely perfectly engineered our digestion is, right down to precise chemical bonds being flexed by specialized proteins. But how delicate is the balance of human life, really, if a single tiny environmental shift in your body's temperature or pH could permanently break down this entire miraculous, microscopic factory? Think about that for a bit. Thanks for exploring the hidden mechanics of digestion with me today. Keep wondering and keep learning.